So it's time for the second talk by Guido Barbujani from the University of Ferrara, Mismeasuring of Man 30 years later. Please, Guido.
Okay, thank you, Giorgio, and thank you all for coming here in this beautiful Venice morning. As a matter of fact, Ian Tartasol gave part of my presentation earlier on, and I'm grateful for that, because this way I will be able to focus more on what happened in the last 30 years after the publication of this book. And as you will see, the story is rather intriguing, and one that Stephen Jay Gould might have liked to write about, because at the same time, certain ideas gained support in the area of human diversity, evolutionary genetics, but at the same time, the opposite idea, that the right way to describe human variation is by the race concept, is still very strong in medical research and still attracts funds and people work in this area. So these are the two sentences I wanted to start from, and you will recognize the second one. You have seen it just 5 minutes ago. When Steve Gould wrote this book, he had two strong ideas in mind, I think. And one is that we are not predetermined entirely by our genes. And the other one is that it's a good thing to measure man. It's a good thing to try to investigate human diversity. But very often, the methods and the approaches that have been developed to describe human variation have turned into a means to rank peoples and group of people, invariably to demonstrate that white men are at the top and other groups, other racial groups, because here we are speaking of races, other races are at a lower level, both from the physical standpoint, from the intellectual standpoint, and ultimately that human society is a fair reflection of a biological gradation of qualities. To illustrate these principles, I am using a more recent paper by Steve Gould. This was published in the collection called I Have Landed, and I apologize for using the Italian figures. I didn't find the original ones. But here you see a portrait of Blumenbach. We will get back to him later on. He proposed a 5-race classification system of humans. Blumenbach was the man who invented the word Caucasian. In his opinion, the first humans came from the Caucasus, they had white skin, and they were beautiful. And as time progressed in two directions, towards Africa and towards Asia, there was a decay of qualities demonstrated by the reduction of beauty in the skulls of these people. Later on, a German anatomist, Tiedemann, decided to do what was done usually at that time, to measure the size of these skulls, thus inferring the amount of brain contained into them. And this is the average he found in Caucasians, so Europeans. Malay are really Australian, people from Australia, Americans, Mongols, and Ethiopians, which was the word he used to describe Africans. And here you see that the cerebral mass is maximum in white people, minimal in black, with the other groups in between. Now, once again, Steve Gould got back to this data, and in his paper, he also published the whole range of the observations for the 5 racial groups studied by Tiedemann. And as you see, there's a slight difference in the averages. These are the averages that here seemed so spread. But on a broader scale, you see that there is an almost complete overlap of the brain masses. No statistician would ever try to differentiate these 5 groups, so there is no difference here. And the important concept here is that the variation within each of these groups is much, much larger than the difference between the averages of these groups. And this concept will come back again several times in my talk, and comes back actually in most studies, most serious studies on human diversity. Now, these kind of observations are the observations that make it very complicated to find a stable catalog of human races. The historical attempts have been many, many. And they all started from the observation that we are all different. And in fact, we are all genetically different. No two of us, except for identical twins, are genetically identical. More than that, by looking at a person's face, very often we get some information on the geographical origin of this person. So it's very clear that this man is more or less from Europe. This man is a Han from China, the broadest human population, 1 billion members. This girl is from Africa, and so on and so forth. But there are two problems here. The first one, and it turns out to be the smaller one, is that some people's aspect is not so clearly informative about their origin. Here you see a dark-skinned girl who everybody would place someplace in Africa. But no, she comes from India. She comes from the Andaman Island, and there is an area of Southern Asia in which many people look like her. And this lady here, my students generally tend to put in Thailand for reasons I don't really understand, but might have to do with the cap she wears. And this is interesting because it means that when we are trying to classify members of our species, we are very sensitive to some cultural messages, and we tend to confound them with the biological messages we are receiving from the people's aspect. But I was saying that this is not the main problem. The main problem is here in this room, and it is the fact that within the same population, people can be very, very different. And because of this internal difference, you will not be surprised if I tell you that among the many, many, many systems of classification of human races proposed through the course of history, the only thing in common is that each of them contradicts all the others. Here is a summary of a few of them, eliminating all the production of crazy people who managed to publish a book sometime in the past or in the present. And I think it's fair to start from Linnaeus, who had 6 races in mind, but 2 of them were really strange. So I'm not going to talk about them. But the main 4 races by Linnaeus are derived from the 4 elements of classical Greek philosophy: air, water, fire, and earth. Europeans are air, if you are interested in this. And then later on, Buffon, who had traveled in Northern Europe and Asia, proposed that also Tartar and Laplanders deserve the status of race. And then came Buffon, then came Kant. This is Immanuel Kant, the philosopher, who had 4 races in mind with this strange Kalmuk race. For some reason, he thought that Kalmuk people were very different from anybody else. Then here you have Blumenbach, and this is probably the most popular way to classify people, 1 race per continent. And then Cuvier came and said, no, there are only 3 kinds of people: white, yellow, and black, and so on and so forth. The main trend here is that the number of races increases. And the reason for that is that as travelers traveled, as explorers explored, they came back to Europe or to the States with new kinds of people who could hardly be fit into pre-existing races. And so the number of races had to be expanded until it reached, in some systems of the 20th century, figures around 200. So it is clear that when you have a system of classification with 200 different items, it's very, very hard, if possible at all, to define clearly, scientifically, the boundaries between these groups. There were a few attempts to reduce the number of human races in the 20th century, but this system was clearly showing some weaknesses. And so around the 60s, some anthropologists began to suggest that perhaps the best way to understand human diversity is not to list the human races. Just as an example of the persistence of these concepts, very recently, an American statistician called Risch very vocally defended the validity of the concept of race in science. And in 2 pages of the same article, he used 2 different racial classification systems. So one of the 2 reasons, we will come later to the second one, but one of the 2 reasons why it seems very hard to use race for a scientific study of humans is that there has never been any agreement on human races. on which are the human races. As most of you know, the seven dwarfs do not exist, but still they have properties. We know that there are seven, and we know their names, although I cannot tell you their names in English because I know them only in Italian. Human races, we don't know how many they are, and we don't know their names. So in strictly philosophical terms, their ontological value is currently less than the value of the seven dwarfs. But I was speaking about people who didn't like the idea of races and began to propose different ways to look at human diversity. One of them is Frank Livingstone, who in 1963 wrote an article with a very, very clear title on the non-existence of human races. And what Livingstone was saying is that he doesn't deny that differences, biological differences, exist among members of the human species. But the point is that these differences cannot be summarized by the discrete packages labeled races. His position could be stated, in other words, as there are no races, there are only clines. Human variation is gradual. Different genes tell us different stories on our evolution. Finding a discrete classification of humans is not the right way to understand the processes underlying this pattern. Livingstone's proposal, of course, wasn't accepted immediately, and actually we can say now that the debate is still going on. One of the people who didn't like it was a very famous geneticist, Dobzhansky, who a few years later replied with a book called Genetic Diversity and Human Equality. And Dobzhansky made it very clear from the beginning that he was not supporting a racist view of society. He said that we all have the same rights, not because we are genetically identical, but because we are all human. But, he said, there are valid races in men. He acknowledged that these races have not been identified yet. But he was confident that with the new methods of biology that were being developed in the 70s, we would soon begin to describe them and define them. So the good news is that the future has come. We live in what, for Dobzhansky, was the future. And in what follows, I will give you 6 little pieces of evidence concerning the issue that Dobzhansky and Frank Livingstone were discussing, but not before having shown you that, as Ian Tattersall anticipated, the race concept is extremely successful in current scientific literature. I did a PubMed search last week, and I found more than 150,000 papers, mostly from the US medical literature, using the word race, or found with using the word race. And you see here, the idea is racial differences in breast cancer risk, racial differences in non-Hodgkin lymphoma, and so on and so forth. So we should try to also to ask ourselves why this concept is so successful. And here is one of the most of the strongest statements of the race concept. In Neil Rish's paper, he published this tree, calling it the evolutionary tree of human races, no citation, no evidence, and he says, okay, we are like this: Africans, Caucasians, Pacific Islanders, East Asians, Native Americans. These are the human races. A few pages later, he forgot this tree and he used a different tree. So how come that these contradictory statements are taken seriously in science and actually inspire hundreds of thousands of new scientific papers? That's the question. But to put things in their appropriate context, let's ask ourselves, let's try to define what we are talking about. Let's try to define genomic differences. So the first thing we can say is that in principle, each cell of our bodies contains the same amount of genetic information, the same text in our genome, 3 billion plus base pairs in its haploid version. So all cells of our body, unless we have a cancer, unless something happened in these cells, should differ by 0 nucleotides, 0 bases. And the same is true of two identical twins. There are no genetic differences between them. when we pick two of us at random, even from different places on Earth, on average, the difference is in 1 out of 1,000 nucleotides. And this has been repeatedly shown. And when we compare instead one of us with a chimp, the difference is between 10 and 30, and recent estimates tend to increase this difference. So until a few years ago, we thought that we were 98% chimpanzees. And this is also the title of a book by Jonathan Marks. Now that copy number variation and other areas of the genome have been studied more thoroughly, we think that this difference is a little bit bigger. And because everything is related with everything else in the world of evolution, there are also relationships between us and vegetables. And I took from Telmo Pievani's presentation this idea in the Rome exhibition, and my estimate is that we differ by 650 out of 1,000 nucleotides. You may correct me if I'm wrong. So we have a 35% of genes that are the same in all living beings for the very good reason that our cells are the same in all living beings, more or less, and have to do the same things. So what I will be talking about from now on are subfractions of this fraction. So this is an estimate of the global human diversity if we take two people at random from this room or from different continents. As you will see, there isn't much difference. Then you expect to find one difference out of 1,000 nucleotides that you consider. And I will give you just 5 or 6 pieces of information, not more than that, because we don't have much time. This is the way evolutionary biologists represent. If I must say, I see that you laugh, but by the end of this slide, I think I will demonstrate that there is no reason to be happy at all. Believe me, believe me. Anyway, this is the way we represent differences between individuals or DNA sequences. And in this case, we are talking about X chromosome DNA sequences in a study published like 10 years ago by Svante Pääbo's group. And here you see that orangutans are on one side of this evolutionary tree with their two subspecies, Sumatra and Borneo, very well differentiated. Here there are clear races or subspecies. And then there are two long branches separating the African apes from the Asian apes. And here we have gorillas, chimpanzees, and bonobos, who are now recognized as an independent species from chimpanzees. Here we have humans. But mind you, this very, very little knot comprises 70 people from all over the planet. So we are so close to each other, so similar to each other, indeed so similar to this specimen whom I chose here, that we can be hardly taken apart. We are very, very similar. The levels of internal diversity of our species are by far the lowest among primates. Not only that, but as you will see, these small differences are accounted for largely by differences between populations, not individuals. In other words, each human population contains a very large share of the global species variation. And this was shown first by Richard Lewontin, who in 1972 published his very famous paper on the apportionment of human diversity, in which he showed that if you divide human genetic diversity in 3 components, one representing differences between members of the same population, the second one representing differences between populations of the same continent or race. In that paper, he used a 7-race classification system. And the third one, representing differences between races. The variation within populations is by far the largest component of human diversity. So people would expect here to find the figures around 20 or 30. He showed it was 85%. Many of us, the oldest I think, may remember that this result was met with disbelief. Among other things, Lewontin was accused to be a Marxist, which was probably true, but had nothing to do with the subject at stake. And an error was found in his statistical treatment of data. But a few years later, an Australian anthropologist reanalyzed the same data, adding another locus, genetic locus, correcting the error, and as you see, the results are almost identical. Then the criticism was that these figures had been estimated from protein data. At that time, it was extremely difficult to work on the large scale using human DNA data. But a few years later, with Luca Cavalli-Sforza, we redid the analysis on a completely different set of genes and individuals, and the results are surprisingly similar to those previously obtained. And in following years, they have been repeatedly confirmed until 2 years ago. This is not a mistake. Lee and collaborators analyzed hundreds of thousands of markers in a paper whose data have been are not considered reliable by everybody. So let's put it this way. It's very difficult to check this many data. But admitting that the error is minor here, once again you see the same figures. And in practice, these figures, 85, 5, and 10%, representing respectively the amount of the general species variance within populations, between populations, and between races or continents, has been confirmed independently many, many times. This means that if the whole population of Earth gets extinguished, but only one single population remains, we will save approximately 85% of the global diversity of the species. In fact, a little bit more if the surviving population is from Africa, a little bit less if the surviving population is from Europe, Asia, and certainly less than that if it is from Australia or from the Americas. An interesting implication of this result comes from recent studies of the entire genome. This is an area that's blossoming. 2 years ago, when I wrote a review paper on this, there were only 16 complete genome sequences. Now we are approaching 1,000. And what I would like Steve Gould to consider and write a paper about is that this enormous increase of the information available has not been accompanied by a comparable increase of our ability to understand biological phenomena. We are investing enormous amounts of money in typing DNA, but I don't think we are understanding much more than we did 2 or 3 years ago before this data were available. I see that Marco agrees with me. Anyway, here you have at least 2 celebrities and the third also important biologist. You recognize here James Watson, the discoverer of the double helix structure of DNA. James Watson stated that our species needs to investigate the genetic basis of the genius, and to do that, we need some genius donates his own DNA, and this is what he did. And so we have, I'm serious, I'm not joking. And so his DNA was typed almost entirely, not entirely because he didn't want to know a few things about potentially pathological genes. A few years later, Craig Venter, you see here, didn't even have to donate his DNA because he typed it at home in his company. And to show that these people have no ego problems, Venter called his sequence the human reference sequence. So people tend to be very confident in this area. A few years later, another scientist, a Korean this time, his name is Kim, but all Koreans are called Kim, so it's useless to give you this piece of information, also completely sequenced his own DNA and published this figure, which I think is very telling on patterns of human diversity in our species. The area of overlap represents a number of DNA polymorphisms shared by the 3 individuals, 1,200,000, so on and so forth. Venter and Watson, as you see, are both US scientists of European origin, and they look like Europeans. And they share 460,000 variable sites, SNPs. A SNP is a technical term, which is less than either of them, Venter and Watson, shares with Kim. So the 2 extreme individuals here are Watson and Venter. Kim is in between, and this is a way to represent their differences. Mind you, this doesn't mean that all Europeans are closer to Koreans than they are to other Europeans. But it does mean that each population is so variable that 2 of us can easily be more different than one of us and a Chinese, a Moroccan, a Venezuelan, you name it. And not keeping this in mind would lead to bad evolutionary inference, I think. A few more things to explain why things are so. This graph is rather difficult to interpret, but it's really like the graph I just showed you. It shows the areas of overlap between DNA variants found in 5 continents. So the blue area represents DNA variants found in the 5 main continents. And these are 81% of the total DNA variants. So in our genome, 81% of the variable things are present at different frequencies, of course, in all continents. That's why, so far, it turned out to be impossible to draw clear lines between the genomes of people in different continents, because we have the same things. It's like the ABO blood group. It's cosmopolitan. It's distributed all over the world. Only a small fraction of DNA variants is continent-specific. 0.06% in Europe, 0.06% in the Americas, 0.06% in Oceania, a little bit more, 91% in Africa. So this is the first sign that Africa is special from the genetic standpoint, and it's different from the other continents. We have other evidence that Africa is special among continents from a rather old paper. 10 years in this field are many years. But here you see the levels of overlap between Europe, Asia, and Africa for long stretches of DNA. You might say, okay, the single DNA polymorphism is not very informative, but if you take many of them at the same time, then you may find better geographical specificities. And this, in a sense, is true. Before I showed you a figure around 80% here. Now it's 50%. But what remains constant is that just a small fraction of human genomes is typically European or typically Asian. A larger fraction is typically African. So in principle, this figure tells me that there are 2 kinds of human genomes. Some human genomes are specifically African. Some human genomes are generically human and are present in all continents, including Africa. These, I think, is one of the most interesting pieces of evidence that we have on human diversity. A couple more things. These 2 people, whose name I don't even try to pronounce because they are some people, and the language includes a number of strange sounds called clicks. These 2 people have a strange privilege. In the moment in which this paper was published, so 2 years ago, I don't know about more recent papers, these were the 2 most different humans studied thus far. So in 2 areas of the Kalahari Desert in Namibia, a few hundred kilometers apart, there are 2 hunter-gatherers who go around, hunt, and live more or less in the same environment, in the same place, and their difference is greater than any genetic difference ever measured in our species. So Africa is really special. Africa contains a broader catalog of human variation than any other place in the world. And finally, this is a bit more complicated, more sophisticated, but I think it's very important. A few years ago, new methods were developed to simulate evolution at the genetic level and then to compare observed data with the results of simulations generated under different models. And this comes from the laboratory of one of the best experts of this field, Laurent Escoffier in Switzerland, and he compared 3 main models of human evolution. You have heard something about them in Ian Tattersalls' talk. Here you have in all these graphs Africa, Asia, the Americas. The present time is at the top, the past is at the bottom. And in this model, as you see, an originally African population gives rise to another population that goes to Asia, and then from Asia a group goes away to the Americas. And these populations are connected by gene flow. This is what has long been called a multi-regional model of human evolution. And by contrast, there is another model whom no doubt Ian Tattersall will like much better, in which an African population gives rise to an Asian population, but as you see here, there is a discontinuity. Current Asians are just Africans who moved into Asia and replaced completely the original population, and then some of them moved to the Americas. And then there is a third model with a small amount of exchange between the archaic and the modern Asian populations. And by simulation, Laurent Escoffier's group demonstrated that this model has a 78% likelihood when compared with real actual data on these populations. And this model prevails 20% of time. whereas this one is not supported by his simulations. So once again, there are many reasons to believe that Africa played a crucial role in human evolution, both from the anthropological standpoint and from the genetic standpoint. So how did all this come about? I'm doing something I shouldn't be doing now. I'm going to summarize human evolution in 4 slides. So don't do it at home. It's not the right way to deal with the things. But in a sense, it's what us scientists do all of the time. So we try to summarize complex phenomena into simple models. And if they are reasonable, then we will understand something that by looking at the whole picture, we couldn't exactly grasp. So I'm showing to you what I think might have happened in the last 100,000 years to our ancestors. Each of them is represented by a spot of a different color to indicate that there was some degree of genetic differentiation at that time. Of course, the world was not devoid of populations. Here we had Neanderthals, here we had Homo erectus, here Floresiensis, perhaps other human species that we haven't described yet. But if we believe Laurent Escoffier's simulation, all of our ancestors, or almost all of them, were here. And their life was tough, because these people couldn't produce food. So they had to walk around, to wander, collecting vegetables whenever they found vegetables they could eat, killing animals, and moving from one place to another when there was nothing to eat in that place. And in the course of this process, they began to explore their continent without knowing where they were, of course. And here you see that the number of humans has increased, and also the number of colors. We know that there is a process called mutation, continuously generating new variation. And as they proceeded in their wandering in Africa, some of them came very close to Palestine and happened actually to cross into Palestine. Then when they cross into Palestine, things change. First of all, the environment is less hostile. There are more resources. The population is very, very small there. There are a few Neanderthals, but Neanderthals were good guys. We eliminated them very fast when we moved into their territories. So something that Darwin had predicted happens here. The infant mortality goes down, and so there is a greater number of children who reach reproductive age and can generate other children who have a higher chance to reach reproductive age. So the population expands. But this increase in population size is an increase of the children of the orange and yellow people who just happened to move here, whereas the blue people or the purple people remained in Africa because they were not in the right place at the right time. And at the end of the story, 30,000 years ago, when almost all of the world is peopled, and everybody in the world is like us, anatomically modern humans, you have in all continents, including Africa, the descendants of the orange and yellow people who made it to Asia and Europe, plus here and there some occasional new colors due to new mutations. Whereas in Africa you have the same people plus the people who didn't go away. Of course, I'm not claiming here that this is all that happened to humans in 100,000 years. But if this reconstruction is not too far from what actually happened, we can make sense of all the data I showed to you earlier on and of many other data I didn't have the time to show to you. Now, do I have 5 more minutes? So if I have 5 more minutes, I will show you why I think that medical doctors should keep this data in mind and take them very seriously, which they generally don't, especially in the US. And I will use an example from the newly developing field of pharmacogenomics. We all know that the same drug at the same dosage has different effects on different people. And generally, if you treat a group of people with the same diagnosis, with the same dosage of a drug, there is a number of people who respond positively, and so they feel better. But there is also a class of people who shows no response. They can take the drug, but nothing happens. And a group of people, generally small, but generally present, who have negative side effects, and so they feel worse after taking the pills. This story has been known for many, many years, and what we know now is that genetic polymorphism affects this response to drugs. And we also have an idea of the general principles of the thing, even though the details are extremely complicated and different for different classes of drugs. But the idea is that there are three phases in the fate of a drug within our body, and the drug here is represented by these pink balls. In the initial phase, some transporter genes produce some proteins that carry the drug to the target. And this is the least interesting part of the thing, because nothing much happens here. We are almost all identical. And then there is an interaction with the surface proteins in the cells produced by genes that we call target genes. And pharmaceutical companies have been investigating this phenomena in extreme detail. But what really matters for the phenomenon I'm describing now is here. After a while, some other genes called DME, drug metabolizing enzymes genes, produce proteins that lead to the catabolism of the drug. And there is polymorphism in these genes. So in some cases, this process is very fast. In some cases, it's lower. Generally, drugs are experimented on a random sample of the population, and so the dosage that we take represents more or less the average reaction of the population. But people who are faster than average don't leave the drug in its place for a sufficient time for it to act. And people who are slower than average leave the drug for too long here and may have side effects. Now, pharmaceutical companies have been investing billions of euros to develop specific drugs for specific markets. And market here means race. If there are different races in different places, then you can sell different races, different drugs. And this even led to the development of a new discipline, which is called racial medicine, rather successful in the US. Now, racial medicine has no possibility whatsoever to work. I understand that this is a strong statement, but I feel like making it. And here is a small demonstration. This is a study of one of the most popular drug metabolizing genes, CYP2D6. And the experiment is very simple. You take a number of people, you give them a certain amount of a chemical that represents the drug without the clinical effects. And then you collect in the urine of these people after a certain amount of time the drug, and you see how much of it was metabolized. And if you put on graph people, there are people who after a while have metabolized a small amount of the drug, so they are called slow metabolizers, people who have been faster and people who have been very fast. And this is a study between Chinese and Swedes, published more than 15 years ago. And as you see, there's a slight difference. The Swedes tended to be slightly faster on average than Chinese, as shown by their histograms, which are shifted to the right with respect to the others. But what is very important here is that in both populations there is a complete range of slow, intermediate, and fast individuals. So there is no hope to be able to develop a drug that will work in Chinese and not in Swedes and vice versa. Whereas it is highly possible, and I think we should expect this in the future, to develop individual drugs. Drugs that work if you are slow or normal or rapid, no matter whether you are from China, from Sweden, or from any other country of the world. And so I will finish by saying that we know that we are all different. Refusing the validity of the concept of race as a valid descriptor of human diversity doesn't mean to say that we think that we are all identical. No. One difference per 1,000 nucleotides over the 3 billion nucleotides of our genome means that each of us differs from any other person by 3 million nucleotides. It's a lot. So these differences do exist. Gorillas are more different than we are. Chimps are more different than we are. And this has some evolutionary relevance. I won't talk about that. But denying this evidence leads to poor evolutionary inference and to errors in medical practice. Now, I'm totally aware that by saying here that we shouldn't be talking about races, I won't influence at all the usage of the word in our common discourse. Races are certainly part of our psychological and social world, to a greater or lesser extent, and certainly to a very high extent in the US. And they will not vanish just because a population geneticist is telling you, okay, forget about it. But on the basis of what we know, and we know a lot in this area, the idea that these concepts refer to a biological reality is devoid of any support. And if things will change in the future, we will be ready to reconsider our opinions. If not, I think we should consider that races at present are not a valid way to describe human diversity.